system for sure. And then if we still have time, we're going to discuss the laboratory portion of the exocrine system. Okay? That's why we have 21 slides. Actually, it should be 22 for pancreas. Oh, yeah. Okay, let's start with the liver. Okay, just remember that your liver is surrounded by capsule, and they are divided into lobules. And under scanner view, you can identify a liver lobule. Although it's not clearly demarcating a lobule, but you can identify that the center is occupied by the central vein, and the periphery is occupied by marked by your portal areas. Okay? And then, if you go into low power, you can identify also the parenchyma or the characteristic cells. Characteristic cells that are arranged in cords or in rows, so you also call them as hepatic plates, and they are separated in between by your sinusoids. So you can see the hepatic plates. In between the spaces are your hepatic sinusoids. And remember that hepatic sinusoids is lined by endothelial cells, and they are associated by macrophage. Okay, now the macrophage are commonly irregular shaped cells wherein you can identify them found also in your sinusoids. Okay, these are the nucleus. This is your nucleus of your one copper cell. Not this one, probably this one, but this one is also your nucleus of your one copper cells. Okay. Now, again, going back to the basic, your hepatocyte is polygonal in shape, mononucleated, or you also have two nucleus in one cell. So what else? Acidophilic cytoplasm, etc. Okay? So you have to go back to the basic. And then also you have to identify the portal area. Let's try to see a bigger and better portal area. Let's go back. Wait. Okay, this is one sample of your portal area, wherein you can identify the portal vein, carrying unoxygenated but nutrient-rich blood. You can see the prominent tunica media layer of your hepatic artery. And then here is your bile duct, which is lined by simple columnar epithelium. But let's try other areas of the portal. Ito. Yan. Yan. So this is your portal vein. This one is your hepatic bile duct. Wait, I'll check first. Ay, pangit. Ayaw. No, not good. Probably that's the cause of his death. Not having good cross-section of the portal area. Still, a little better. So this one is your portal vein, bile duct, hepatic artery. You can see the prominent tunica media, right? Okay, so that's it for your liver. Now for gallbladder, you have to identify, aside from, you know already the lining epithelium, which is simple columnar. This time, you're going to identify all the layers of your gallbladder. So of course, aside from the function, which is storage of your bile and concentration of your bile, this is scalar view. have to identify the layers. Which first is mucosa. The mucosa is comprised only by two layers. You have the lining epithelium and the lamina propria. Okay? So, oh, sorry. Posing muna. Posing. Wait. Posing. Yan. Here. It's better. Okay, this is better. Posing. Because this is your lining epithelium and this is the lamina propria portion. Now, found in the lamina propria is this structure, which is the characteristic feature of the gallbladder, located in the lamina propria. This is your RA sinus. Spell RA sinus. Right? Or Rokitansky-Askov sinus, so you better spell that correctly. Okay, so that is mucosa. And remember, there is no submucosa. Next is already your fibromuscular layer made up of your elastic fiber and your elastic and smooth muscle. That's why it's fibromuscular. The fibrous portion is the elastic and the muscular portion is the smooth muscle. So that's why it's fibromuscular layer followed by a thin perimuscular layer. And then you have here, let's check if that is only adventitia. There is that's serosa. Serosa because it is bounded by your simple squamous or mesothelium. Okay, it's covered by a peritoneum, but in some areas where it is associated by liver, oh, this one's the liver, and this one is the gallbladder. There's no serosa in between, but only adventitia. Remember, I told you that the side of the gallbladder attached to the liver, there's no serosa, but instead tunica adventitia, right? And the other side, exposed to the peritoneum, the outer layer is tunica serosa. Okay?
Doc, excuse me. Would you please go back and show us again the RA sinus?
That is a sample of your RA sinus. It's an invagination or crypts that is also an RA sinus. Actually, when you identify RA sinus, it is just an invagination of your mucosa. That's why if it is an invagination, the lining epithelium is similar to the surface epithelium. Right? So this is your RA or Rokitansky-Askov sinus. That's another RA. Also an RA. You have two RAs. So that is a characteristic feature of your gallbladder. Okay? So once you see the RA sinus, you know it is gallbladder. Okay? Pancreas. So under the digestive system, this is the one that secretes pancreatic enzymes, but actually the enzymes are secreted as proenzyme, meaning inactive, and they are just activated once they are found inside the duodenum. Okay? And they open into the second portion of the duodenum. So there are two portions of your pancreas. You have the pancreatic acini for the digestive system, and your pancreatic islet of Langerhans, that is for endocrine function of the pancreas. So that is your scanner view. Now, by scanning, you try to identify portions of your pancreas where you can identify the islet to differentiate that from your parotid gland, because that is very similar. They are both serous. So you have to identify for your islet of Langerhans. So commonly, that's your islet. That's why small islands, which is discovered by Langerhans. It's an endocrine portion. So this is your islet of Langerhans. That is mostly your pancreatic acini, which secretes your enzymes. Now, each pancreatic acini is surrounded by acinar cells. Surrounded by acinar cells that are pyramidal. So this is one acinar, pancreatic acini, etc. Now, individually, you have the acinar cells, the secretory cells, and you can see or identify the nucleus at the center of the lumen or at the lumen, which is light-stained nucleus, actually your centroacinar cells. Okay, which is also characteristic for your pancreas if you compare that with your parotid gland. Okay?